Ever wondered how scientists know exactly what's in a glass of water, or the food on your plate, or even the air you're breathing right now? Well, today, we're going to pull back the curtain and uncover the secrets, starting from the very bottom, from the atom all the way up. So how do they do it? The answer is this really remarkable journey, one that takes us from the totally invisible world of atoms all the way to the powerful tools of modern analysis. Our first stop on this journey, the atomic blueprint. Look, before we can analyze anything, we have to understand what it's made of, right? That means we have to start at the very, very beginning with the fundamental building blocks of, well, everything. And this idea isn't new at all. We're talking over 2,000 years ago. The Greek philosopher Democritus had this thought. What if you kept cutting a piece of matter in half, over and over? Eventually, you'd hit a point where you just couldn't cut it anymore. He called that final, tiny piece atomos, which literally means indivisible or uncuttable. And it turns out he was pretty much spot on. Today, we know the atom as the basic unit of any chemical element. Everything you see, everything you touch, it's all made of these tiny, tiny particles. They are, quite literally, the Lego bricks of the entire universe. What's so fascinating is that these atoms aren't just some random jumble. They're organized into the periodic table, which is like a master blueprint that reveals all these predictable patterns. Take atomic size, for instance. You'd think as you move from left to right, adding more stuff, that atoms would get bigger, right? But nope, they actually get smaller because a stronger nucleus pulls the electrons in tighter. Then, as you go down a column, you're adding new layers, new shells, and that makes the atoms get bigger. Kind of cool, huh? All right, so we have our building blocks, we have our Legos, but how do they actually connect to create everything from a simple drop of water to a complex protein? Well, that's all about molecular architecture. Atoms have two main ways of sticking together. First up, you've got the ionic bond. This is where one atom basically just gives an electron to another. This little transfer creates opposite charges, a positive and a negative, and they attract each other like tiny magnets. The other way is a covalent bond, which is less of a transfer and more of a partnership, where atoms share electrons. And the way these atoms bond actually determines the molecule's final 3D shape. And the rule for this is incredibly simple. Electron pairs can't stand each other. They all want their own personal space, and this pushing and pulling forces the molecule into a very specific shape. This repulsion creates all these different distinct shapes. Methane, for example, forms a perfect little tetrahedron. Ammonia is almost the same, but it has an extra pair of electrons that pushes the other bonds down, creating this kind of pyramid shape. Others, like formaldehyde, end up totally flat. And believe me, this geometry is everything. It dictates how molecules act and react in the world. Okay, so we know what atoms are, we know how they build molecules. But this brings up a huge problem for chemists. I mean, how on earth are you supposed to count particles that are way too small to see and exist in numbers you can't even imagine? You use a really, really big number. This number right here is Avogadro's number. It's a standardized amount, kind of like a chemist's dozen. But instead of 12, it's 602 sextillion. It's a standard unit that lets us count atoms on a scale that actually makes sense. And we call that chemist's dozen a mole. A mole of anything, whether it's carbon atoms or water molecules, is always 6.02 times 10 to the 23rd particles. And here's why that's such a game changer. The mole is the universal bridge. It connects the microscopic world of atoms that we can't see to the world we can see and measure in a lab. And this, right here, this is the key. A mole of water, a mole of glucose, and a mole of table sugar all have the exact same number of molecules. But because the individual molecules are different sizes, their masses are totally different. So you gotta remember, a mole is a count, like a dozen. It's not a weight. That distinction is absolutely critical. So now we can count molecules. That's great. But in the real world, things are messy. Most samples are these complex mixtures. So how do we sort through all that and isolate the one thing we actually want to study? To unscramble that big molecular soup, you basically make all the different components run a race against each other. This race is called chromatography. You load your mixed-up sample, and then a fluid, called the mobile phase, starts to push everything along. And here's the really clever part. As they move, different molecules interact with the material they're passing through differently. Some get slowed down, they get a little sticky, while others just zoom right on by. This difference in speed is what separates the mixture into all its pure individual parts. All right, we are on the home stretch. We've counted them, we've separated them. Now for the final step, identification. How do we look at something pure and say with 100% certainty, yep, that's caffeine, or that's definitely chlorine? Well, the answer lies in light. See, the bonds inside a molecule are kind of tuned to absorb very specific energies or colors of light. They'll soak up the colors they like and just completely ignore all the rest. And because every single type of molecule has its own unique structure, it has its own unique pattern of which light it absorbs and which it lets pass through. This pattern is as unique to a molecule as a fingerprint is to a person. Seriously. And in chemistry, we call that molecular fingerprint a spectrum. By shining light through our sample and seeing which colors get absorbed, we can identify exactly what molecule we're looking at. But it gets even better than that. Thanks to something called Beer's law, we can't just identify the substance, we can figure out how much of it is there. The law basically says that the amount of light that gets absorbed is directly related to the concentration. So more molecules means more light gets blocked. It's really that simple. So let's bring it all the way back to our original question. How do we know what's in your water? Well, we start by understanding the atomic blueprint. We use the mole to count the uncountable. We make them run a molecular race to separate them out. And finally, we use light to read each molecule's unique fingerprint. And that is the incredible journey, from the invisible atom to the undeniable power of modern analysis.